Hello, this is a brief overview of your immune system or immune responses. The immune system is what helps defend us from infection. Something that causes an infection is called a pathogen. A pathogen is an infection-causing or a disease-causing uh, substance, and gen means to generate, and pathos is talking about feeling, or in this case, pain. So there are three layers to your immune response, and they are uh, basically in order of how something would pass through them. The first one are barriers, things like your skin, the oils on your skin, which are bactericidal, or just like the skin itself, which is stuck together. Your sweat and saliva, your gastric juice, every surface of your body that is exposed to the external environment, including the inside of your lungs, right, your respiratory tract, is lined with something that can help to fight off an infection before it begins. So like the mucus layer in the inside of your respiratory tract with the ciliated hairs that can sweep things away, that's part of it. And the barrier is probably the most important part because if something doesn't get in, then there's no infection. But if something does get in, say we have here a splinter coming into the skin, bringing with it whatever's on the splinter, which is going to be some living things for sure. We have non-specific responses. These happen no matter what. These happen even if this was a completely sterilized thing and you weren't ever going to get an infection. So we have, for the non-specific responses, some uh, plasma running outside of your capillaries nearby there, being let out, which increases the temperature and the pressure in the area. Temperature and pressure, inflammation of that area will help to push things out from whence they came, or I guess whence they came if I were being grammatically correct. And macrophages, so big white blood cells, come as well, which start to eat anything unusual that they find, and they identify what's unusual or not based on the name tag, the antigen, which is present on the outside of those cells. So this is the ID card or the name tag or something that says, this is who I am. So if this is a cell that's entering your body or any cell in your body, it will have markers on the outside called antigens. There may be one type, and there's probably going to be lots of types of markers on the outside of that cell. Your immune system has learned to recognize your cells as being harmless. Anything different must be, in the brain of a one-celled organism, must be bad. So if they see an antigen they do not recognize, a macrophage will eat it. This is non-specific. It doesn't matter if they've really seen it before, if it's dangerous, not dangerous. This is going to happen. The macrophage then calls over a series of cells, starting with helper T cells, to begin the specific immune responses, which has the most number of steps. That's the part you're probably going to want to study the most. So we're going to go through this process here, but first I want to make sure antigen is the marker on the outside of a cell, any cell, and antibodies, these are the goal. These are what we are hoping to produce through this whole process. If we can produce antibodies, then we have success. We can successfully fight off this infection. So something comes into your body, we're going to call it a pathogen because it's potentially infectious. Maybe you recognize it and you already know how to make antibodies against it, in which case this whole procedure would be cut short. But we're going to pretend like we have a new thing. We've never seen this pathogen before and we don't know how to fight against it. So it enters the body by breaking through your skin or some other external barrier. Right away, just because of the injury, you will have inflammation, swelling, and macrophages go to check what's going on. If they find anything that they've never seen before, those macrophages will eat. Remember, eating, cell eating is called phagocytosis. They will eat that bacteria, fungus, virus, whatever it is. When they eat it, they will display on their own surface. They will rip off all of these, I guess they're these ones, all of these antigens. As they digest this cell, they kill this cell, they will rip off copies of those antigens, those name tags, and display them on their external surface. That's what's happening in this animation down here. A macrophage is eating some kind of pathogen, digesting it, killing it, and the markers that were present on the outside of the cell are then copied to the outside of the macrophage cell. This is like a trophy. Look what I killed, and it will call over a specific type of cell called a helper T cell. So you have a macrophage that eats some pathogen. It calls over a helper T cell. Helper T cell shows up here in this diagram. And that helper T cell's got a couple of jobs. Number one is to check. Are you overreacting? Have we seen this before? Is it harmless? Just, is there really a problem? And if it hasn't seen it before, then the helper T cell's job is to very clearly scream for help. And it can't scream with sound, right? It can't make sound. Sound is pressure waves that a single-celled organism just can't do. But it can release messengers into your bloodstream and into your lymph. And those messengers will go throughout your whole body calling for help. And two types of cells will come over to help. The two types of cells that come over to help, I'm just going to zoom in on the chart over here, are called T cells and B cells. And T cells and B cells both have different jobs to do. So the helper T cell is a type of T cell that calls for help from both of these guys, the T cells and the B cells. You'll have some T cells that come, and we're going to call these ones, instead of cytotoxic T cells, we're going to call them by a, I think, more fun name. We're going to call them killer T cells. And their job is to hunt down any of these pathogens they can find and stab them. They have little pockets of digestive enzymes inside, and they can use those digestive enzymes, inject it into the cell wall or the cell membrane of something else they find, and cause it to be digested. They can kill things very, very quickly. Some of these killer T cells will stick around long term. Lots of T cells have a very short lifespan. As you might imagine, it's a pretty violent life. But some of them will stick around and become memory cells, which help to remember this particular pathogen and how to defeat it. On the other side, we have the B cells. Sometimes you say beta cells. I say B cells. And the B cells have also a couple of fates that they could end up doing. One of them, we're going to skip to the part where a memory B cell. A memory B cell sticks around later, does the same job as a memory T cell, and the memory B cells and T cells help to inform your immune system how we beat this thing last time. But the stars of this whole show are called plasma cells. And what plasma cells do is they learn from the, what's going to show up over here, the antigens that are captured, oh, missed it, by the macrophages, read by those helper T cells to make sure they're a threat. The plasma cells will learn exactly how to make a copy of something called an antibody. So I'm going to write here, plasma cells have the biggest job, which is to make antibodies that stick to just one antigen. They don't stick to any of your helpful antigens that are on your cells normally. That would be called an autoimmune disease, where your body is thinking that its own cells are invading. Instead, they create an antibody that just sticks to that one antigen and no others. This is tricky to do. It takes them a while to learn how to do it. But once they can do it, they can create it's not countless, you could count them, I suppose, but really, really, really large numbers of those antibodies and send them out all through your blood and through your lymph. When those antibodies attach to an antigen of a pathogen, there are two basic fates. The one we're going to focus on in this course is called agglutination, where these antibodies, once they stick to those antigens, become sticky, gluey. So agglutination is a term that happens when those sticky things cause the pathogen to stick to other pathogens or to other things in your body, maybe just the wall of a skin cell that's nearby. And they can't go anywhere and they can't do anything because they're stuck. The other thing that's related is called neutralization, where they make enough of a barrier around the entire pathogen that it just can't reach the outside world. It's just like you put it in a bubble and it can't help. So the overall process here was you have some kind of infection, something pokes through your skin. You have a non-specific response where a macrophage comes, eats that thing, and displays its antigen or marker on its cell membrane. A helper T cell comes over. The helper T cell has a job to figure out, does this thing really pose a threat? It'll call T cells and B cells. Here we're just looking at B cells. The T cells could be killer T cells, which have the job of doing murders. And we have the B cells, which can become plasma cells. And plasma cells are the most important type of immune cell that you have. Plasma cells can make these things called antibodies. Antibodies are tiny little proteins that can stick to antigens, and once they do, they can cause agglutination, where those pathogens can no longer do their job of causing you trouble. Both the B cells and the T cells can have memory cells, though, that stick around, which makes it easier to fight off this infection the next time you have it. So I have a list of key terms here. Make sure you can define all of these. This concept, especially the antigen-antibody relationship, is going to be very important when we talk about blood typing as well.